Good morning students. In this video class, we are going to discuss plastid and the structure of chloroplast. Plastids was first discovered by Haeckel, but the term plastid was given by A.F.U. Schimper in the year 1885. Plastids are normally found in all green plants. They are also found in certain protists. For example, Euglena. And they are also found in few algae. Now, Schimper classified plastids into three categories based on their structure, function and the pigments found in them. Plastids are classified into three types. They are leucoplast, chloroplast and chromoplast. Now, leuco basically means white or colorless, and these plastids are normally found in parts of the plant where they are normally do not expose themselves to the sunlight. That means they are found under the ground. For example, tubers, root and rhizome. Tuber, example, potato. Root is tapioca. Tapioca is nothing but tapioca chips, that is tuberous root, which is colorless, and rhizome is banana. The real stem of banana is under the ground called rhizome, which is also colorless. So the function of leucoplast is storage of food material. Now the next one is chloroplast and chromoplast. Both chloroplast and chromoplast are colored plastid. Chloroplast are green colored plastids. They have green, they are called as green plastids because they contain a pigment chlorophyll in them, which gives green color. They are basically found in all the leaves, and in certain, when the plant is young, even the stem will have chloroplast. The function of chloroplast is photosynthesis. Now the next one is chromoplast. They are also colored plastid, but other than green. They have red color, yellow, orange and so on. They are found in flowers, that is in petals, fruits and in certain variegated leaves of ornamental plant. Their function is to attract the insect for pollination. So this is the classification of plastids given by Schimper. Now let us understand the structure of chloroplast. Now we are going to see the structure of chloroplast. Chloroplast is a green plastid. So it contains a pigment called chlorophyll in them, and normally the shape of the chloroplast is either oval or discoidal in higher plants. Whereas the shape of the chloroplast in some algae which you have already studied, it can be either cup-shaped in Chlamydomonas, star-shaped in Zygnema and so on. But we are now concentrating only on the chloroplast found in the higher plants. The shape of the chloroplast found in all higher plants will be either oval or discoidal in shape. So the structure of chloroplast is either oval or discoidal in shape, and they are the diameter of this chloroplast is 2 to 10 millimeter in diameter and 1 to 2 millimeter in thickness. Now you are able to see the structure. I have drawn the outer membrane. So this is the outer membrane of the chloroplast, and they also have one more membrane, that is the inner membrane. So it is a double membrane bound organelles. So chloroplast is a double membrane bound organelle. This is the outer membrane and this is the inner membrane. And between the outer and the inner membrane, there is a space found, and this space is called as intermembranous space or periplastidal space. So the space found in between the two membrane is called as intermembranous space, or otherwise it is called as periplastidal space. Now this outer and the inner membrane together is called as an envelope. Now inside this outer and the inner membrane, you will be able to see a gelatinous matrix made up of lipoproteinaceous substances called stroma. So inside this envelope, it is filled with a gelatinous matrix made up of lipoproteinaceous substances, and this is called as stroma. Now let us see what are all the things that is found inside the stroma. Now inside the envelope, stroma is present. What are all the things that is found in the stroma? Stroma consists of DNA. They have got a circular DNA. So inside the stroma, you will be able to see a circular DNA. Since chloroplast contain their own DNA, it is otherwise called a semi-autonomous organelle. So stroma consists of DNA. They have got ribosomes, and the ribosome is of 70S type. The ribosomes found in chloroplast is called as plastidal ribosomes, and they are of 70S type. They also have enzymes necessary for starch synthesis. It is not protein synthesis, but it is starch synthesis. Stroma consists of lot of enzymes which play a very important role in the dark reaction of photosynthesis. During dark reaction of photosynthesis, enzymes play a very important role, and one of the important enzymes found in stroma is rubisco. Rubisco is nothing but ribulose 1,5-bisphosphate carboxylase, which is the most abundant protein found in the world. So stroma consists of DNA, ribosomes, enzymes like rubisco, which play a very important role in starch synthesis. Now on the stroma, there is some invagination like this, and this invagination has number of membrane which are stacked one on the other. So these structure together is called as granum, or singular granum. So along with DNA, RNA, they also have RNA, ribosomes, enzymes. You have an invagination which leads to number of membrane structure which are arranged one after the other like a pile of coin. Here also it starts from the inner membrane, an invagination begins, and then a stacked membranous structure arranged one after the other. So this is called as granum. And how many granum will be there inside a chloroplast? Normally there will be between 40 to 60 grana formed per chloroplast. Normally this granum is connected to the inner membrane, but they are also connected to each other. Between one grana to other grana also they are connected. So the structure which connects one grana with that of another grana is called as frit membrane, or otherwise it is called as stroma lamella. So this structure is called as stroma lamella, or otherwise called as frit membrane. Now I am going to take this granum outside for a better understanding. So this is how the grana are arranged one after the other like a pile of coin arranged one after the other. So each structure, this is the entire structure of grana or granum. Individual structure is called as thylakoids. So this is one thylakoid, this is another thylakoid, this is another thylakoid. There are many thylakoids arranged one after the other to form grana. Now I am going to take this one thylakoid. If you take one thylakoid, it will be like this. So this membrane will be having a lumen inside. So this is how they are arranged. And on this thylakoid membrane, many pigments are found. Pigments like chlorophyll A, chlorophyll B, chlorophyll C, xanthophyll, carotenoids, and all the pigments necessary for the light reaction of photosynthesis is found in the thylakoid or the grana. So if you are drawing like this, actually the thylakoid membrane will be having a lumen. On the outer side of the lumen will be having number of pigments, and these pigments, all these which I am drawing is pigments. Pigments play a very important role in capturing the sunlight and converting the sunlight into chemical energy. So all the pigments are found in the granum or in the thylakoid membrane. So a light reaction of photosynthesis takes place in the thylakoid membrane or in the grana, and dark reaction of photosynthesis takes place in the stroma. So in this class we have understood the structure of chloroplast and the classification of plastids in detail. Thank you.